Yes.
Okay. So the topic of this lecture is regulatory aspects of comparability exercise. I am going to give a short regulatory history about comparability from the FDA perspective, EMA, and ICH guideline. The first draft guidance by the FDA about comparability was issued in February of 2003. Draft guidance comparability protocols, chemistry manufacturing controls information. That was focused on biologics mainly. And it described what is needed in order for a company that makes changes to a biological manufacturing process in order to introduce those changes during the life cycle of the product. In 2016, April 2016, a new draft guidance was issued, which the title of which was expanded to Draft Guidance Comparability Protocols for Human Drugs and Biologics, Chemistry Manufacturing Controls Information. This draft guidance was finalized to be a final guidance in October 2022. And that guidance title is Guidance for Industry Comparability Protocols for Post-Approval Changes for the Chemistry, Manufacturing, and Controls Information in an NDA, ANDA, or BLA. And what I am going to highlight are the differences between the FDA and EMA, and also how do they refer to the ICH guideline. As you see here, the 2022 guidance discuss the option of submitting comparability protocols also for drugs in NDAs or ANDAs, and not only for biologics. The history in EMA is a bit different. In 1998, the first concept paper on the development of CPMP guidance on comparability of biotechnology-derived products was issued. That concept paper turned to a guideline in December 2003, as guideline on comparability of medicinal products containing biotechnology-derived proteins as active substance, non-clinical and clinical issues. This guidance talked both on the development phase of a product and also of the post-approval phase of a product, and also discussed the option not only of introducing changes for a product in a facility by a sponsor or by the manufacturer, but also the option of what we call today biosimilars product. So that was the first difference between the approach of the FDA in comparison to the approach of the European authorities. That they already in December 2003 included the option of having what we call today biosimilar, meaning manufacturing the same product in a different facility, even by a different developer. In July 2007, there is a new guideline, which is the guideline on comparability of medicinal products containing biotechnology-derived proteins as active substance and non-clinical and clinical issues. And that one excludes the biosimilars because in 2005, the development of a new guideline, which is focused on biosimilars, was started in Europe, so they excluded the biosimilars from the comparability guideline, which is the current one. The ICH guideline, which is the ICH Q5E, in November 2003, there was the first version for release for consultation, which is the comparability of biological products subject to changes in their manufacturing process. And in November 2004, this guideline was approved by the steering committee, and then it was accepted and implemented by the different countries. And that was the basis of the comparability guidance that I just mentioned by the FDA and the EMA. And we will see what are the principles, the scope of each one of the guidelines, and by that we will see what are the differences between them. The FDA guideline scope is for comparability of human drugs, biological products subject to NDA, ANDA, and BLA before and after manufacturing changes. The fact that they added the NDA and ANDA is something that happened only recently, as I mentioned before. Prior to that, it was required only for biotechnological and biological products. And I must say that in our experience as a consulting company, we always recommend to manufacturers of drugs which are not biological or biotechnological to implement the principles of the comparability protocol, because it is one of the most efficient ways to ensure that the manufacturing changes don't have any negative impact on the product after the change. The FDA guideline refers to drug substance or drug products. Its purpose is to provide evidence that the manufacturing process changes will not adversely impact on the quality, safety, and efficacy of the drug. And the one point which is highlighted in all guidance is that the products don't need to be identical, but they should be highly similar. And how you show that it is highly similar, it is by determining your acceptance criteria a priori. I will touch on that when I talk about the protocol, the comparability protocol, and I am sure that Andre will give you some examples of that in this session. EMA, the principles, the scope of the EMA guideline are for assessing the comparability of biological products before and after manufacturing. Those changes are during development or post-approval. Initial versions included also biosimilar products, as I mentioned, and when biosimilars guideline was developed, biosimilars were excluded from the comparability guideline. What you can see here is that this guideline does not address the option of comparability protocols for drug products other than biological. It doesn't mean that you are not allowed to implement those protocols, but it is not a requirement. It's not a regulatory requirement. For a biological, biotechnological, ATMP in Europe, it is a requirement to use the comparability protocol for any change that you intend to make in your process or in your product. It relates to drug substance or drug products, similarly to the FDA. And similarly to the FDA, the purpose is to provide evidence that the manufacturing process changes will not adversely impact quality, safety, and efficacy of the product. And again, not expected to be identical, but highly similar. The scope of the ICH 5E is somehow covering both options, but it is more similar to the EMA guideline. The principles for assessing the comparability of biological products before and after manufacturing, changes where manufacturing changes are made during development of the product or in the manufacturing process of products for which a marketing authorization has been granted. I just want to mention here that the FDA guidance talk only about post-approval changes and not during development, although it is expected that you use a comparability protocol for biological and biotechnological products also during development. It relates again to drug substance and drug product and provides the evidence, and you need to provide evidence that manufacturing process changes will not adversely impact the quality, safety, and so on. And the last point, similarly to what I mentioned already. The general principles of the comparability exercise is the demonstration of comparability does not necessarily mean that the quality attributes of the pre-change and post-change product are identical, but that they are highly similar and that the existing knowledge is sufficiently predictive to ensure that any differences in quality attributes have no adverse impact upon safety or efficacy of the drug product. This point is really important because at the beginning, when they started to talk about comparability, that was the reason for which the authorities were against the biosimilar concept, because they said that only the developer and manufacturer of a biotechnology or biological product has enough knowledge in order to assess whether the products are comparable or not. I think that the only reason for which they agreed at the end of the day, both the EMA and the FDA, to extend it to biosimilar is the development in the analytical area. Because today we have analytical tools that can really show the comparability of products. And on that, Andrea, of course, will talk in a few minutes. Determination of comparability can be based on a combination of analytical testing, biological assays, and in some cases, non-clinical and clinical data. If a manufacturer can provide assurance of comparability through analytical studies alone, non-clinical or clinical studies with the post-change product are not warranted. Based on our experience, in most cases, comparability can be assured by analytical testing and biological assays. PK in animal models may also be required. In rare cases, preclinical or clinical studies are required. And if we talk about biological and biotechnology and ATMP products, if the analytical testing is not good enough in order to determine comparability, and PK model doesn't give a straightforward answer, then it most probably will be required to conduct clinical studies, because preclinical, non-clinical studies, animal studies are not good enough in order to predict the clinical outcome. It is, of course, a regulatory requirement, but when we are talking about comparability, it is less relevant to do a toxicology study rather than to do maybe a PK clinical study in order to show the comparability. Comparability protocol should include scope of change, current process versus process post-implementation of the changes, risk assessment of the proposed changes. If there are a few changes during the process, then each one should be evaluated and the risk of that change to the final product should be assessed. number of batches to be used for comparability exercise. It's not only the number of batches, but the batches to be used prior to the change, and those to be used after the change should be determined in the comparability protocol. That is very important in order to show that those were not picked because of their high quality, but those are for instance, the 3 last batches prior to the change and the 3 first batches after the change, so that you can show that the protocol and the results are not biased. Acceptance criteria for the analytical comparability should be determined in the protocol, not after having the results. And there is a decision tree for comparability assessment that following that tree, you can decide to which extent analytical comparability needs to be executed. Are PK studies needed? Are clinical studies needed? Based on the results of your comparability exercise. And the principles of that are the minor changes in quality attributes with minimal potential of clinical impact, and also if that happens in the early stages of the clinical development. In those cases, most probably that only analytical comparability will be sufficient, and maybe you will need to do some non-clinical PK studies. If major changes in quality attributes with considerable potential clinical impact, and if you are at advanced stages of clinical development, in those cases, non-clinical PK or clinical PK studies may be required. And as I mentioned before, based on those results, it might be that some clinical studies may be required. What is certain is that if you introduce changes during the conduct of a phase 3 study, you will need to do some clinical comparability because you are really at the end of the development. And if you want to rely on all the data that you have gathered until that point, you need to show that your changes did not change the product to a level that you cannot rely on all your development history. And that is really important. And it is important in terms of the decision when to implement a change. It might be better to implement a change as a post-approval change than implementing it prior to the approval or during phase 3. Consultation with regulatory authorities during development phase. It is highly recommended to ask for regulatory advice before the execution of the comparability protocol and implementation of the change. If you are approaching FDA, then during a type C meeting or any other type B meeting, you can provide the outline of the comparability protocol and get their concurrence with that. And if you are submitting that to the European authorities, then either by scientific advice with EMA or national authorities, that is the way to address it and get their advice. And approval of comparability protocol of post-approval changes. That is something that is also recommended. The EMA and FDA would accept to get a comparability protocol for post-approval changes as part of the NDA, BLA, or MAA. So when you submit your registration file, if you submit a comparability protocol to cover any potential post-approval changes, then you may get, with the approval of your product, you may get also the approval of the comparability protocol. That means that when you implement that protocol, the results of that, the report can be submitted and will be reviewed in a shorter timeline as long as it is according to the protocol that was approved by the authority. And for the FDA, that may change from post-approval submission, which is 6 months, to a CB30, which is only 30 days or maximum 60 days. And by EMA, it may be classified as a variation type 1B instead of type 2. Again, that is subject to discussion with the rapporteur, with the authorities, in order to get that agreement. So I've finished my part, the regulatory introduction. I thank you, and I invite Andrea to continue with his presentation.
Thank you very much, Rivka, for your lecture. Just to remind everybody, Andre, just a second, please. If you have any questions, please write them down in the chat and we can address all of the questions at the end. I see that a hand was raised. You can ask it orally at the end of the lectures as well if you prefer, but if it's easier for you, you can simply write it down. And please, Andre, go ahead and.
Thank you, Rivka, for this fast forward retrospective history and setting the regulatory framework for today. And thank you, Roy. So my name is Andre Abts. I'm working at Protagene, a global leading analytical CRO. Protagene was founded 25 years ago. We have 4 sites, 3 in Germany, in Dortmund, Heilbronn, and Heidelberg, and 1 site in Boston, Massachusetts. We offer as a CRO solutions for CMC and CGG projects in all aspects of analytics. We support our clients in very specific questions which require a deep analytical expertise on the one hand, and on the other hand, we are also supporting entire developments from really early stage, for example, clone selection, up to market approval. You're responsible for the entire analytics in these cases, and we are also responsible for the release and stability testing, even when CMOs are involved. So my background, I'm a chemist by training. I have a PhD in biochemistry in the field of antibiotics. I'm working at Protagene since around almost 10 years. Again, the deep CMC understanding through leading several NBE and biosimilar programs from early development up to market approval. And currently I'm heading the global PM department for all CMC projects. And today, I will guide you through the second part of the webinar. I will show you some analytical strategies, how to perform comparabilities, as well as some examples for pre and post approval changes, as well as for biosimilarity data. So when we're talking about comparabilities, we need to first define what is the API, the drug substance. And we have always, when we're talking about biologics, but also on small molecules, the drug substance is always a mixture, and we have a certain degree of heterogeneity. And this heterogeneity is basically defined by process variability, as well as by the variability of the methods, which can detect the certain, the certain amount of species. And as Rivka has already pointed out, the manufacturer needs to define the pattern of heterogeneity, needs to demonstrate consistency of these different lots used in preclinical and clinical trials. And this is typically captured by the specifications we define and also by the stability studies. And it is very important, and I want to highlight this again. In a typical development, we have a lot of changes introduced to different stages. So when we start in a preclinical environment and we go to the phase 1 or phase 3, typically we have some changes. I don't know, we have manufacturing process optimizations, we have changes in the media, or we have some other changes. And that is very important that we align the time point when we perform the comparability exercises to be completed when we approach our dossiers, when we approach our IND for phase 1, for phase 3, that we have already completed the comparability to ensure that we take the information gathered from the preclinical and approach into the phase 1 clinical stages, that we already can confirm that the material is comparable to each other. So that is the first step, basically, where we need to have this comparability exercise. Next one would be to approach from phase 1 to phase 3. If, for example, we have an upscaling or other differences and changes introduced into the manufacturing or into our product, to show again that the phase 3 and the phase 1 material is comparable to each other. And again, if we go to the BLA, MMA, or to commercial, we also may have some transfer of the process and have some other changes to the molecule. Again, also here, we need to ensure that the material is comparable to the other materials used and that everything is comparable to each other. So when comparabilities can occur, so as already shown and discussed, so there might be some in the DS, for example, we have some USP or DSP changes. We might change the host expression cell line. We might have some changes in the modifications in the cell culture. We might change some formulation or filling of the DP. We might need to improve our manufacturing process by increasing scale or stability of the product. Sometimes also needed by regulatory requirements to compile with these new requirements to do some changes. And also if we transfer the manufacturing process from cell line development up to commercial scale at the end, then also here changes occur. And sometimes even nowadays, the raw materials might change and we need to also react on this one. And this is just a very brief outline what Rafka already has mentioned. So we have this ICH Q5B. In principle, we compare, and we will show you some examples later on. We can compare the post-change product against the pre-change product on DS or DP level. We will evaluate the potential adverse impact on DP. And finally, we need to keep in mind to ensure quality, safety, and efficacy on the DP level. So selection of analytical panel is difficult. It needs to be selected very, very thoroughly. I mean, in our drug substance, as already pointed out on the first slide, we have a lot of different materials and ingredients in there. So we have, hopefully, in most of the cases, a very pure API. But sometimes in biologics, we also have some isoforms of that. We might have some degradants to a certain extent, and we also have from the production, from the host cells, we might have some DNA residuals in there, some protein residuals, and also some components or remaining components from the media. And also we need some detergents to keep our API happy in solution, and hopefully not, but it might also that we need to test for microbials in there. And for all these kind of different ingredients, what we have in our drug substance, we need to find and identify some suitable analytical methods to verify that we have the content as well as the shape under control. So for the API, for example, we are doing the classical analytical characterization for aggregation by ZQV and CGE, charge variance by XCIF, hydrophobic interactions with RP and HIC, and also primary structure with peptide mapping. Then regarding the host cell related species, we can do some ELISAs and MS to determine the HCP content. We can verify the residual DNA by qPCR and also analyze by some chemical methods the amount of media components left in the drug substance. When we're going to formulation and process-related species, we need to have a look at the detergents, leachables, extractables, but also in amino acids and saccharides to determine the content in there and determine, in case, some certain limits if they are toxic. And then, of course, we need to show microbial testing that we are on a safe side here, by endotoxins, bioburden, and sterility. At the end, we really carefully need to select the appropriate analytical methods to identify often small differences in the quality attributes, since the changes might introduce only a few percentage of change, which might then have a high impact on the efficacy later on. And the characterization message, this is a requirement, needs to be scientifically sound for the intended purpose, which is a very big discussed topic. So in general, they say they should be qualified. And if we have release testing methods already available, then these should be validated. But not also, we need to have a look at the methods. We also need to have a look at the certain samples. So if we here, for example, have a typically manufacturing process shown exemplarily, there are certain steps where different IPC samples can be analyzed and needs to be analyzed, because if we are looking at differences in the host cell proteins, for example, we cannot analyze the DP before and after the change, because in the DP, the host cell proteins are no longer, or hopefully should not be longer available. So we need to have a look at the IPC samples basically to determine the levels of host cell proteins at a certain degree. And the same holds true for aggregation, for example. We need to show, or we can analyze different kind of IPC samples to see that the aggregation can be removed or controlled at different kind of levels. And also leachables can be monitored typically at a DP level. And this is also that we not only need to select the certain analytical methods, but we also need to show authorities that we have our process under control and that we really can analyze the correct samples to verify the differences in the change we introduced. Finally, we can use the tiered approach for comparability assessment, and that is also part of the comparability protocol. Basically, that is also what Rivka has mentioned. So you can start with the first step, which is an analytical evaluation, where you do the typical QC test results. You do an extended analytical characterization if this is needed. You can, or if sometimes you need to show also the degradation and extended temperature is needed, and also to determine the process of impurities here. If this shows some differences, you can go to the next level to say, okay, then I need to look at the biological characterization or the functional assays. So I can look for potency, I can look for ADCC, CDC, and also for FC effector functions, binding. And if this even shows some differences, then you go to the next level, which is then basically the animal PK studies or PK/PD studies in rodents or in primates, which is then the next level. Hopefully you could already show in the first two steps that your process did not change any critical differences to the molecule in terms of safety and efficacy, because afterwards, if you're going to animal PK or PK/PD study, then the effort, and of course also that will impact the project highly, to introduce then clinical PK and comparability studies, or even clinical experience or efficacy studies, which then makes it very, very complicated and should be avoided if as far as possible. So now I will, so just a very brief summary of the first part. So comparabilities can or will occur throughout the entire life cycle of a development program. It is really suggested to align the timing with the regulatory filing to have the comparability completed at the certain levels and certain gates. The selection of the analytical method panel is dependent on the impact and basically based on the risk assessment, as well as the correct samples needs to be selected to really analyze it at the correct level. And of course, it's recommended to use this tiered approach. We're going now to some case studies. I will have found some literature. Of course, most of them are not public, but I found some literature of a group where they compared some already market approved products before at certain expiry dates, and then they could see that some manufacturing changes might have happened in the past. So I will start with NBEL etanercept, which is a dimeric fusion protein, which consists of two parts. On the one hand, we have a TNF alpha binding domain, and we have an FC part of an IgG1 antibody. On both parts, we have a highly glycosylated structures. So we have O glycans and N glycans in there. And the glycans are also very important. This is why I'm pinpointing this here out for the mode of action of the molecule. And what you can see here on the slide is basically the bright blue is the pre-change and the dark blue is the post-change glycan profile. And you see that the G2F glycan is reduced drastically after introducing a change. So here you see two clustering, and so here on the x-axis you have the expiry date of different lots. So we have a G2F level of around 50% before the change, and after the change was introduced, we have a drop by 20% to a level of 30%, which is basically relatively drastic. And in addition to that, these batches show also a change in the charge profile. So here you see a CAX analysis. So before the change, the amount of basic variance was quite low, and after the change, the basic variance was quite high. So here we have a change from 15 to 30%, up to 40 to 60%. And so this might be related to the C-terminal lysin variants, which are not mode of action dependent, but which might happen in antibodies in the processing of that. Finally, these changes also is approved change by the authorities, so this is acceptable. So we assume, or the authors assume, that there is no impact on mode of action, on safety or efficacy, and the product is still marketed under the same label. One additional example was the ARANESP, so the darabopruitin alpha, which is a human analogon to erythropoietin, so to EPO. This product differs to the human EPO, where it has more N-glycosylation sites, and the mode of action of the protein here is dependent on the number of and the isoforms of the sialic acids on the glycans. And here you see a capillary zone electrophoresis analysis, which is able to distinct or separate the different isoforms, which is related to the different sialic acid contents. And what you see in the pre-change here in blue in comparison to the post-change in red is that the isoforms sialic acid, the amount of sialic acid is reduced in the post-change. So you see here it's was higher sialylated than the red curve, which was lower sialylated. But even here we have a change of around 10% of one of the species. We have a very distinct group. So it is also an approved change. And indeed here it could be figured out that in 2008, the EMA approved a major change in the manufacturing process with an extensive comparability exercise. And this change was approved, and still here the efficacy was not impacted by this change of the sialic acids. Another example on an already marketed product is rituximab, an IgG1 monoclonal antibody which targets CD20 markers on the surface of cancer cells, and it's also involved in rheumatoid arthritis. And here it's an antibody, so also here it's important to have this Fc effector functions, which are often related to glycosylation. Here we have some differences between the pre and the post change. Again, antibodies tend to have this basic variants focus on C-terminal lysins or glutamine on the N-terminal. So here in the pre-change, the amount of basic variants was between 30 and 50%, and after the change was introduced, it was really reduced up to down to 10%. And in the glycan analysis, it's only a small change here in the G0 glycan, which was increased after the post change. And you also see this here clearly, that also here is a group of increased G0 glycans. It's only around 1% increase, but the increase of G0 glycans causes also an increase of ADCC, which is also here crucial for the mode of action. Then this is directly related. Also the ADCC data, which are not shown here, showed an increase by a factor of 3, but still this was approved by the agencies, and the overall efficacy of the molecule did not have a significant change. And so this is also still, or was still manufactured and was approved and still sold under the same brand name. So these are just 2 examples about some marketed products. And now I will just spend the last 5 to 10 minutes on the biosimilarity aspects of one molecule. So we have, just to summarize it, so alterations of the glycosylation profile might happen. They are often related in terms of antibodies to the mode of action. We have seen some changes in the N and C terminal heterogeneity and some variation in ADCC activity. But nevertheless, the differences are most probably caused by a manufacturing change, because the glycosylation profile is often dependent on the cell line, on the growth conditions, on the purification sequences, what you have in your product. All product testases remained on the same label on the market. And so this indicates that there were no big differences in terms of safety and efficacy, and thereby all these changes were accepted already by health authorities. And now I will move to the biosimilar slides. So we are going to see some biosimilars of Humira. So here we have the example of adalimumab. So the Humira is the originator was AbbVie, and this study, which you will see in the next slide, it's from Amgen, which have proposed a biosimilar. Actually, that was approved already in 2016 as the first Humira biosimilar. It's called Amjevita in the US, and actually they entered market now in January 2023 in the US. The study was conducted of the analytical methods shown here. So we have 22 structural assays applied, 17 functional assays, and 7 general assays here. So in total, the entire biosimilarity session, which is also a kind of comparability actually, because you compare the originator against the biosimilar candidate and need to show that they are comparable to each other. All of the methods were qualified or validated depending on the levels. In this biosimilarity approach, they used 10 biosimilar candidate lots, which are representative of the final product. It used 18 European lots of Humira and 24 US lots of Humira, and they sourced the originators over the time span of 5 years. Not all of the assays were performed for all assays. It was dependent on the criticality of the assays as well as on the availability of the material. But the FDA confirmed the statistical approach, and so they could continue with this strategy. I will just focus on three aspects here. So what they did basically is the biosimilar candidate, which is called from Amgen ABP 501. They have a different formulation than the originator from AbbVie. And what you see if you analyze the Humira formulation and the ABP formulation, you see basically that the same product formulated in different formulations has lower HMWs in the biosimilar candidate formulation than in the originator formulation. And this is actually a problem because even you have lower HMW levels, you are not comparable. You don't have the same levels as the originator. So what they did basically is that Amgen rebuffered their own product into the originator formulation, and they executed a degradation kinetic study over a certain time of months under 50 degrees, and they could show that the slope of the biosimilar candidate in the Humira formulation was the same as the originator in the same formulation, that they can really show that the degradation kinetics is comparable. In this study, they used to justify that they have in principle in the formulation a difference in the HMWs, which is not related to safety, but the molecule behaves the same in the same formulation, that they have comparability here. Another example in this study was the KEX analysis. So here you can distribute the molecule in terms of acidic, main, and basic variants, what you can see over here. And what you actually also see here in the graphs is that the basic variants, so the red dots here are always the biosimilar candidate. In blue is the US marketed Humira samples, and in green the EU Humira samples. And you see the basic variants are lower and outside of the corridor of the originator. So you also see this here, that this peak is decreased in terms of the other samples shown over here. And they did an extensive characterization of this peak here, and they could definitely show, and this is also related to the other antibodies, it is the amount of C-terminal clipping is different, but since the C-terminalizing is not involved into the mode of action, it was a justifiable differences between the originator and the biosimilar candidate. Another example is here the glycosylation, and glycosylation is critical in Humira, since we have Humira or the adalimumab is binding the soluble TNF alpha, which you will see later on. It's is dependent on the action, on the mode of action, and by binding the soluble TNF alpha, it reduces the inflammation processes, but it's also binding the receptor or the membrane-bound TNF alpha, and thereby the FC effector function of the antibody uses ADCC and CDC to lyse the macrophages and other kind of samples to also here reduce the inflammation processes, and thereby also the glycosylation of the molecule is crucial for the mode of action. And actually what you see here is in the typical glycan pattern chromatogram. And if you evaluate all the different glycan patterns, you see that in the sialylation group, it's higher in the biosimilar candidate than in the originator molecules. And also galactosylation is a little bit higher. So sialylation is critical. It's only it's below 2%. So that's not a problem here, but it would be higher, then it might increase the ADCC activity. So since this is very low, and they performed also an ADCC study where they did not see any differences, this could also be justified in discussion with the FDA. Galactosylation, on the other hand, so an increased galactosylation will cause an increase in CDC activity by binding the C1q. But also here the differences were not that high and significant, and also the corresponding CDC study basically did show comparable data here. And afucosylation and high-mannose, they tend to be lower, but also here both afucosylation and high-mannose are responsible for higher ADCC activities and high-mannose for clearance of the antibody. But also here, and this is now shown on the last slide, is basically the mode of action, and also in the ADCC and CDC activity for the transmembrane TNF-alpha binding. They could show comparability on a high extent, and also on the soluble TNF-alpha binding, they also could show that the proposed biosimilar candidate was in the range of the originator. That finally, of course, the potency is most important. They could show that the efficacy is similar, and I mean it was approved by FDA now on the market, so that's a very good study executed here. So just to summarize this now. So even different formulations between biosimilar candidate and originator were investigated under accelerated stress conditions, and those showed a similar degradation profile in the originator formulation. Difference in charge variance was investigated by extensive characterization of these impurities, and this revealed no impact on potency. And even slight glycosylation differences is not preclude a finding of biosimilarity when potency and PK profile is not affected. And that is also, I want to just come back to Rivka's part, also for the glycosylation pattern, since it is so crucial to mode of action, Amgen needed to show some PK studies actually, because they had some differences in their quality attributes on the analytical level. And also here, just to make it a round story, also for biosimilarity, you also need to impact everything in terms of safety, potency, efficacy, as you do also for an MBE. And that's basically everything. So yeah, so we have 10 minutes left. So we are happy to take any questions or any comments.
And we have a few questions from the attendees in the Q&A. So I think that we can start there. The first question from Martin Said, Mr. Martin Said. Is there a list of approved products?
Yeah, so actually I found that question a bit difficult to answer. If the question is whether there is a list of biosimilar approved products, then it's something that you can get to that list. If you are talking about products that were subject to comparability exercise and they are approved, then I can tell you without checking that 99% of the biological products were subjected at some point of time, either whether during development or post-approval, to a comparability protocol, because that is the way that that you develop the product. So I answer your question, that would be great. If you meant something else, then we can address that after the meeting.
Yeah, so I just can comment on this. So Riff, you're totally right. I mean, every product will go in the comparability sooner or later in the development. But if you want to have some literature examples, it's really hard to find, because of course, the manufacturers and the developers do not share their comparability protocols public. So there are a few literature around which summarizes, as I have shown today, a few lessons learned and a few examples, but not that really much. Yeah.
The next question from Gamzenu Aibar. I hope that I pronounced it correctly. You mentioned that not expected to be identical, but highly similar. Can you elaborate the highly similar? For example, could stability characteristic results and analysis profiles be similar, but not identical in the analytical aspect?
I will give a quick answer and I then refer to Andre because he already provided examples of that. So the idea is that that you determine a priori what is highly similar in your eyes. So when you write your protocol, you need to define what are your acceptance criteria for each one of the analytical methods and for the totality of the exercise. And that will actually define what is highly similar. In practice, I think that the examples that Andre provided actually give you the answer to that. So it is all the characteristic studies, the stability and the analytical profile, all of that together provide you with the answer whether it is highly similar or not.
Yes, totally agree. So I mean, you've put this decision tree in your slides, which basically also gives you an answer to that. I mean, it's a continuous process. You start with a risk assessment. You define which impacts might be critical to safety. Again, I mean, it's very boring, but it's always the same. It's quality, safety, and efficacy. You always need to think about, okay, is this change now critical? In your risk assessment, you will have a certain answer, which assays are really critical. You define your ranges, you predefine your results, and then basically you have already your, as Rivka has mentioned, your classification, if it is then similar or acceptable.
Yeah, and the question of acceptable is really important. A lot of years ago, when I just established the company, it's like about I established the company 14 years ago, or about 12 years ago, I worked with a company that was one of the first to develop biosimilars product, and they provided me with results of their manufacturing, and they had very high molecular weight forms. And they asked me whether they can provide it to the authorities as a biosimilar product. And I said, look, you have high molecular weight forms. That is a risk in the eye of the authorities. All you need to do is to improve your process, and that may result in a more similar product. At that point, I thought that result that they had is not justifiable because that was a safety concern. But and that's the way that you need to assess your acceptance criteria. Can we move to the next?
Yes, thank you. Now we have 3 questions from Mr. Talal Al-Zahaya. If you want to ask them orally, please raise your hand and I'll allow you to speak. I'll start with the first question, but if I see you raise your hand, ask more. Okay, I can see you now, so I'll just allow you to speak. Please go ahead.
Hello.
You need to unmute, please. Okay.
Yeah, is it okay now?
Yes,
yes,
we can hear you.
we
Perfect.
can hear you.
Yeah, thank you very much for such enlightening presentations. Now, my first question was, we always hear about the approval of products without the need for clinical phase 3 efficacy and safety studies. And we've seen something about this in the recently in the FDA and in the EMA guidelines. But my question is, has there been any products so far, biologic or biosimilar, being approved without a clinical phase 3?
So as far as I know, not so far. I think the MHRA, so the UK authorities, they are the leading authorities here in this aspect. Of course, there is a big debate the last 2 years to remove phase 3. I think there is now the first small molecule actually, which is in the MHRA without a phase 3 approved, but I'm not pretty sure. As far as I know, for biologics or biosimilars, not so far as far as I know. I think it will be really tremendous. And the MHRA would expect the other first, and then the other authorities follow this example.
Yeah, because there was a lot of talks about insulins, for example. You know, they are considered as a small biological molecules, and they are very, very simple since they are not glycosylated and there's no post-translation in these molecules. So in one of my scientific advices with the BPharm, actually, they advised to go without, but I cannot see it happening anywhere. They keep talking about it, but I cannot see it happening. When you go to them, they will start stitching their feet and coming out of it. So I wonder if this is going to happen one day.
Yeah, and you are right. That is also our experience. We are working now with insulin-based product, and the company just wanted to change the source of the insulin, the manufacturer from one company to another. Both are insulin, both are USP and EP insulin, but both the FDA and the EMA are requesting a full comparability protocol and the phase 3 clinical trial.
Yeah.
So
I think there must be one big pharmaceutical company to be the braves, to be first with a very, very thorough. I mean, you need a very extended convincing analytical package to really say, okay, I mean, I would say you need to be 90% similar to really convince the authorities not to do the phase 3.
That's true, yeah. You see, I worked with Protein many years ago, and they did some comparability for me on the physical-chemical characterization, and the data was fantastic. But again, when we went to the BPharm and to the EMA, they insisted on a phase 3, and to be honest, we haven't done it yet, but we are doing our best. Now, my second question was just to elaborate for the benefit of all on board. What do you mean by scientifically sound analytical method compared to fully validated analytical method?
Yeah, so basically scientifically, so I mean, it is so from the regulatory perspective, you say that all your characterization methods, which you do not use for release and stability, must not be validated. So you need to show a qualification. This is how we call it in our company, but there's different names for that. But this scientifically sound means that you can really show that your method is capable to show differences. So that you can small differences with your method that is very robust. So basically, you need to show more or less, let's say, 70-80% of a validation study also for a qualification study right now to really say that's for characterization purposes. I mean, of course, it depends. If you have really an impurity method, you really need to show a lot of qualification activities like accuracy, linearity, etc.
Yeah, okay. Now, my last question was about the originator comparability of different batches. You know, most of these biologics, they are either EU marketed or USA marketed or rest of the world marketed. And to be honest, from my experience, the API for all these markets may usually come from one single source. So for example, a multinational, they have their API plant, production plant in Italy, let's say, and the same API is used for the product in the EU, and it's used for the product in USA and maybe for the rest of the world. And when we were doing our work, we were asked to have batches from the EU market and batches from the USA market, and the API, I think it comes from the same source. Yes, the formulation and the finished product may be done in a plant in Germany or done in a plant in the United States. So, have you ever did an exercise comparing different batches with different ages? And with different expiry times, taken from EU and taken from the United States for the originator,
yes,
not for a biosimilar.
Yeah, so we did this. I mean, you need to show basically for your FDA. I think right now the numbers are, you need to have at least 10 EU and 10 US batches from different expiry dates to define a certain corridor of your originator. And it's totally right. I mean, when there's international companies that typically have one DS production site, they're distributed, and then they have several DP filling sites, and then you have an US and EU and the rest of the world DP, which may come from the same DS, but you never know. That is why it's so important to have really sourced continuously in a biosimilar programs the originator molecule. Really over the course of, I mean, they've shown the data from Amgen here, they have sourced it over the course of 5 years. And then you have the chance to have different DS batches related to the different DP batches which you have sourced. So our thumb rule right now is really to have at least 2 months of difference in expiry dates. And it really depends on the availability of the originator molecules on the market and your sourcing agencies and your sourcing partners if they can get them. But you need to have a very good sourcing strategy to try to maximize the difference in the originators.
Yeah, I mean, can you elaborate on the magnitude of differences between different batches of the originator?
Oh, it depends. I mean, we also have seen through a course of 3 years. When you're doing a biosimilar program and you source different batches, you also see manufacturing changes of the originators. We've seen this also in our labs for glycosylation, as I've shown today. We have seen this, and then of course a biosimilar company who wants to develop a biosimilar needs to then think about which of the so typically you take then the newer data, the newest batches to be aligned with them. It really depends. So glycosylation can be a difference of 20%. Yeah, you cannot really say this right now here.
Yeah, I think the biosimilar developers, they are getting a hard treatment compared to the originators, because I am sure many originators on the market, if you want to subject them to the biosimilar approval route, I don't know how they perform.
I don't know. I think that also the originators need to provide the good data and the high quality data, and they need to invest a lot of work until they reach to their product. So, you know, both companies, both kind of companies are subject to a lot of criticism and dialogues with the regulatory authorities.
For sure, yeah.
What I wanted to say something else about that. Okay. About the fact that you need to take examples from the US market and the European market, even if the API is manufactured at the same facility. Sometimes you have different release criteria for one market as opposed to the other. And that also adds additional thing that you need to be aware of and consider in your comparability studies. So that's why you need to, not only because of the formulation, but also because sometimes the specifications from for a batch released to the US and a batch released to the EU are different. And I am sure that Andre has examples of that.
Yeah.
Yeah, but I agree. Yeah.
Okay. I believe that we are out of time. Thank you very much, Talal, for your questions. They gave us an opportunity to elaborate a little bit more and be a little bit more specific. And we are going to send everybody here and everybody that has attended the session a link to the recording of this session, and we'll add the presentations that accompanied each lecture. And please feel free to reach each of us. We'll be happy to continue answering your questions and helping you with your projects. Please, Rivka, did you want to say?
There is one last request here for a session on statistical analysis and CQA determination. I believe that Protogene, together with us, can address that. request as well.
Yeah, that's
And
fine. Happy to do so. Yeah.
we can plan that for the future.
Yes, so please follow us. We give many events and through the Prodigy. So please stay tuned for the next joint event and the other events that we'll put up. And thank you very much, Andre, and for your team. Thank you, Rivka, as well. And thank you all of the attendees that stayed through the lecture. Have a wonderful week.
Thank you all, the ADRES team, and yeah, have a good week.
Thank you.
Yeah,
Take care.
thank you.
Bye.
Goodbye.
Goodbye.